Okay, well, I'd firstly like to thank the organisers for giving me the opportunity to talk to you today. So my lab is based in Oxford. We're primarily a mitochondrial research lab, so we're interested in mitochondrial dysfunction in disease. And for the last few years, we've got very interested in seeing whether cancer cell metabolism might be a therapeutic strategy in trying to tackle those really difficult to treat cancers in patients. So I'm going to talk to you about that work today. This work is unpublished, it's all new data, and I'd like to share with you some of our findings, which I think could be sort of very interesting into the wider community and how we should be looking to do these experiments in the lab. So factors affecting cancer therapy, there are a number: early diagnosis, genetics, the mutation profile, drug resistance are all very important, as is hypoxia. But what I'm going to discuss with you today is our work on substrate availability, primarily glucose. So this work was carried out mainly by Michelle Potter, who was a very talented graduate student in my group, which was finished, and she started this project 4 years ago. A couple of placement students, Yvette Hode and Georgina Dean, have picked up various bits of this work, and I'll talk about their work at the end. And these were placement students from the University of Bath. The work was funded by a kind donation from the Williams Fund, who also are currently funding a couple of projects in my lab, and we're very grateful to them. So, to start really, we got into this area really from some other work we were doing. So we're very interested in mitochondrial associated drug toxicity. It's a big problem for the pharmaceutical sector. There are a number of compounds that show this toxicity that have what are called black box warnings. These are warnings that the FDA give out to drugs which are really at the last chance saloon. Any more toxic effects that could be found with these compounds would lead to the product to be withdrawn from the market. So we're interested in whether some of these compounds that were mitochondrial active could be of benefit in the treatment of cancer. Obviously, these compounds are toxic, so if we're going to use them in patients, we need to find a way of increasing, if we have to increase the dose, to deliver them safely. So we're very interested in taking some of these compounds, when we can identify interesting ones, into a sort of a nanoparticle delivery system. So we work very closely with Dr. Helen Townley's group, who work in engineering, also part of our department, who are developing interesting silica nanoparticles to deliver drugs and other things like RNAi, etc. And Helen's group recently published a paper in nanomaterials, nanotechnology, which is well worth a read, and it details some of the different particles that they have available. So when we started this work, the Hanahan paper had just come out in 2011. Aerobic glycolysis was one of the new pathways, and so we were interested in targeting aerobic glycolysis as a strategy. So cancer cells, as at that time, we thought were primarily glycolytic in nature, and as I just said, dichloroacetate has shown some benefit in brain tumour patients. There was a study by a Canadian group, a couple of papers now, where in very small numbers of patients there seemed to be some benefit of giving the drug in combination with surgery to the patient. So as you can see from the panel on the right, the tumour was reduced. And these were patients that really shouldn't have shown this shrinkage in the tumour. So what DCA does, it actually drives the cells to use their mitochondria. So on the left-hand panel you can see the current way of thinking that tumours are very glycolytic, so the glucose is taken through to lactate and doesn't really go into the mitochondria because of the HIF inactivation of PDH. And by adding dichloroacetate, what you do is you take the glucose through, so it's actually taken through pyruvate into the TCA cycle, and there's various factors that the groups seem to believe are important, so increased apoptosis, proliferation and ROS, and those factors could be important in the reduction in cell growth and actually tumour destruction. So this is where we were. So we started off with DCA and we looked at it in the lab, and we tried it in a couple of models, but essentially the main results from our work in 2D cultures, so these are just flat cells grown in a flask, was that DCA really had very minimal effects. All the cell lines we looked at, there was no obvious increased killing. So you can see on the left-hand panel a brain tumour line where we're looking at the effect of DCA, and really, as you can see, there really wasn't a very big effect, even on quite high doses, 20 millimolar, which is what people have used previously in vitro. But we did see some interesting effects in spheroids. So as you can see on the right-hand side, some of the cell lines responded very well. So at the bottom of this panel, the U87MG, which is a glioblastoma cell line, at high doses of DCA, you see lots of spheroid destruction. You get a very big spheroid at 5 millimolar, and when we take these spheroids apart, they're primarily dead cells. So it was definitely doing something. The RH30 line in the middle, which is a pediatric rhabdomyosarcoma line, you see a shrinkage at the high doses. You can see that quite clearly. And then the RD line, which is also a rhabdomyosarcoma line, showed no real effect at all. So the spheroids grew at pretty much at the same rate. So we were wondering at this point in time, well, why was there a difference? Was it because these cells showed different levels of glycolysis, which is where we thought the DCA was working at this point in time? And so to study this, we've used a platform sold by Luxcell Biosciences, who are presenting their different products here today. So essentially 2 assays. One measures oxygen consumption, which you use a seal system where you put oil on the top and you measure the consumption of oxygen in the media. And the other system looks at the acidification of the media, so how much lactate the cells are producing, primarily an indication of glycolysis. So using these 2 approaches, you can measure the amount of OXPHOS, or mitochondrial respiration, and glycolysis. And so we've done this for different lines, and the results are quite interesting. So if you take cell lines and you grow them on high glucose media, 25 millimolar, which is what most people grow their cells in, you get an interesting result. So we expected to see the cell lines to be very glycolytic, so towards the right-hand side of the graph. Some are, but a number are on the other side, so they're showing fairly low glycolytic rates but high rates of respiration. So the RD cell line, interestingly, was the one that didn't respond to DCA, is quite oxidative. The RH30 is somewhere in the middle, so it's got quite a lot of respiration but also some glycolytic function. Whereas the U87, the one that responded the best, was shown down on the left-hand side, or right-hand side, sorry, which is very, very glycolytic and not doing very much in the way of respiration at all. So to take this further, we're interested in the DCA effects. So what we decided to do was to take the extremes. So we looked at the RD and the U87 MG cells, and then we're developing other approaches with other groups in Oxford, so the chemistry department, to look at what happens to metabolites when we take the glucose through into the mitochondria. So this is a schematic of what we're doing. So we take glucose, labeled with carbon 13 on all the carbons, 6 carbons, and then we track that put that into the cells. These experiments are carried out on high glucose. So what we expected to see was the glucose would be taken into the cell, go through glycolysis, and the glycolytic line U87Mg would basically break down that glucose into lactate, so we get a nice big lactate peak. It wouldn't take the glucose further than lactate or pyruvate, whereas the RD line, which we know has a more OXPHOS phenotype on this high glucose media, would take the glucose all the way into the TCA cycle. So we generate glutamate and also glutamine products, and we can track these using NMR. This is in collaboration with Tim Claridge in chemistry. So when we did this, we got some quite interesting results. So the U87, which is the glycolytic line, is shown at the top. So you can see a lactate peak, quite a big one, bigger than the RD, which is shown down the bottom. What was interesting that both lines had quite a considerable amount of TCA cycle intermediates. So these are the glutamate peaks shown, C2, C3, C4 on the traces. And if we zoom in on those areas, we found something even more interesting. So we go through the RD line first, so you can see the typical TCA cycle intermediate. So you've got glutamate 2, 3, and 4, quite nice, strong peaks. But we see the same type of peaks in the U87, and this is the line that doesn't really do a lot of OXPHOS, so we didn't expect the glucose to be going into the TCA cycle. Here we've got very strong peaks, but also second peaks, there's more doublets formed. Now at the moment we're not quite clear what these doublets mean. Does this mean that the intermediates are going round and round the TCA cycle, and we're breaking down those molecules into smaller pieces? And this is an area that we're looking at in more detail. We're looking at different traces, trying to use glutamine to see where that goes into the cycle. This work is well covered in a paper by Marin Valencia in Cell Metabolism, so it's worth a read and have a look at that. So we've got an interesting thing here. So the glucose in the U87 cells is taken to the TCA cycle, but we're getting very, very low rates of respiration, just about on the detectability. So how is that happening? And we're not quite clear as to what's going on at this point in time. So what we decided to do was to look at these same type of experiments, looking at the oxygen consumption rates and acidification rates, but on media that we thought was more physiological. We all grow our cells at very high glucose concentrations, which makes the cells grow fast, but it isn't really what's going on in the body, where the levels of glucose are probably between 4 and 7 millimolar in plasma, and also much lower than that in tissue, so probably down to the 1 millimolar range. So if you look at the bottom of this graph, first, U87 cells, what you can see quite strikingly is as when we take the glucose down from the high glucose concentrations we normally grow them in, and we do this by incubating the cells overnight in these different glucose concentrations. The cells are quite happy, they proliferate, they grow at the same rate, at least in this short time frame, as they would do on high glucose. And you can see quite clearly on the left-hand panel at the bottom, the oxygen consumption rates increased dramatically with the U87 cells when you had them on low glucose. The RD cells don't show much of a change. They've got a reasonably high rate of respiration anyway, even on 20 millimolar glucose, and it's fairly flat across the graph, so it's not a big change, if any change at all. The acidification rates are interesting, so on 20 millimolar and even 5 millimolar, you see quite high levels of acidification, but when you get down to the low glucose range, 1 in this case, then the acidification levels are very low. So you can imagine in a tissue, the amount of glycolysis going on for these cells, if they were in vivo, would be quite small. So that was quite interesting and quite different from what we expected to see. What we tried to do is to try and look at different lines, so do different lines show profiles. So Michelle's project was really looking at pediatric gliomas to see whether we could look at some treatments were effective on them. So on this graph we're showing the difference between the pediatric gliomas and the adult glioblastomas. So these are quite different tumour types, they're both in the brain, but they have different origins and they're going to be quite different. So if we look at the adult ones first, so that's the U87 and the MO59K, you can see a big increase in respiration as you drop down the glucose concentrations. But you don't see this with any of the glioblastoma lines. The ones on the left are the high grade, these are the faster growing ones, and they look very similar to the RD cells. So from the rate of respiration they have at 20 millimolar is pretty much the same as what it is when you drop it down to sort of 1 or 0 millimolar. Interesting, the UW479 cells have a very fast respiration rates. The low grade pediatric glioma is somewhere in between. It's still more similar to the flat ones of the RD cells, so it's not quite like the adult ones, but obviously the numbers are small. But it's interesting to see now we're getting profiling of different lines, how they respond to our drug regimes that we're going to try. So at the moment we're looking at Ewing's, which is another pediatric tumour type, to see how they profile, and then we'll see how they respond to some of our therapeutic strategies. So at this point in time we weren't quite sure what to do, so we thought that tumours were going to be very glycolytic, and yes they are, some of them when we grow them on high glucose, but when we grow them on more physiological glucose or even down at 1 millimolar, as you can see on this slide, essentially the levels of glucose. So the cancer cells on this slide show very little acidification at all. They're all on the left-hand side of the plot, so showing rates of oxygen consumption. Some are very high on this low glucose, and these are primarily the adult brain tumour lines, but there's a spread. So rather than look at these lines, which are going to be quite heterogeneous in terms of their genetic mutation load, we decided to look at a different system that we thought had a high energy need. So we're looking at a model here, shown on the right-hand side, where we're trying to target the P-glycoprotein. So P-glycoprotein is commonly found in many cancers. It's the multi-drug resistant component which allows those cancer cells to survive chemotherapy agents. Now sometimes cancers can have high levels of this pump initially, or it can be induced by chemotherapy. So the lines we've been using we got from the NIH, so they've been around for a while, and I'll just discuss those in a bit more detail in a second. And just to say that targeting the mitochondria is a very interesting area in cancer treatment in general. So several papers have now come out showing that metformin, which is a commonly used diabetic treatment, reduces cancer risk in patients that take the drug compared to those that don't. How does metformin work? We're not quite sure. It seems to target the mitochondria, so it could be impairing mitochondrial function. And what I'm showing on this side are some of the data. So it's quite nice to see something work. So the idea was that we were going to target the PGP, the ATP component of this channel, which is involved in pumping out the compound. So these cell lines are either the parental cell line, which is the one shown in orange, the KB3 one, or the KBV one, which expresses the channel. And you can see on the top right hand side the V1 expresses this PGP protein at high levels. And if we increase the concentration of a drug that impairs the mitochondria, so we've used a drug called fenformin, which works very similarly to metformin, which is much more potent. On the top part of this graph, as you can see, as we increase the dose of fenformin on different glucose concentrations, the levels of cells drops, so the cells slow down, their growth slows down when you increase the dose of the drug. There's not a big difference between the cell lines, and you see a general trend of reduced growth. But what we see quite nicely on the bottom is an increase in cell death, particularly on the V1 line, and the V1 line when grown on low glucose, which is the glucose concentration we think is more relevant physiologically. So you see a very high rate of death, up to 50% on the higher doses of the compound on the 1 millimolar glucose, whereas you see more death on high glucose, but it's not quite as striking. So obviously these cells aren't as sensitive when they're on high glucose. So we've looked into this in a bit more detail, and what we find is very interesting. So one thing I haven't mentioned yet is that the 3-1 cell line grows very fast. There's no chemotherapy drug on that cell line when it's grown in culture. The V1 has vinblastine included in the media, so that's sort of testing the drug resistance of the line. It grows much slower, so the turnovers are very different. So if you focus on the orange bars first, you can see, if you look at the left-hand side, there's a clear drop in ATP levels, so these cells aren't able to make any energy when they're grown on low glucose with high concentrations of the drug. This is the same at 5, but when you start to look at them on 25 millimolar, then the effects are much less marked. So there's still a drop in ATP. We get this interesting spike at 1 millimolar fenformin. That's real. That's something for another day. It's quite an interesting phenomenon. We see quite a lot with compounds that interact with the mitochondria. The KBV one is quite different. It's not showing such a dramatic change, so the ATP levels don't drop. You do see a drop on 1 millimolar metformin on the left-hand side, but generally there's not a big drop in ATP, even though these cells are dying. So there's no change in ATP, but on the 1 millimolar glucose, as I showed on the previous slide, there's a huge increase in death. And we think this is the difference we're seeing here is partly due to the growth rates of these cells and the demands of the cells on ATP. The 3-1s are much faster dividing, need a lot more ATP, even though the ATP is required for the channel, and that seems to be impaired in the V1s that increases the killing, but doesn't really change the growth. We looked at respiration because we felt that fenformin targets mitochondrial respiration. This is using the Luxcel probes. So again, this data is normalized to the no drug concentration on each of the glucose concentrations, and you can see across the board that there is a drop in respiration with fenformin. So it is impairing oxygen consumption in these cell lines as we anticipated it would. It does it at all the concentrations, so even at 25 millimolar. But as I showed you before, there's not really much of an effect at 5 millimolar, 25 millimolar glucose. So what's going on there? And we've looked into this in a bit more detail. So what happens is that the cells do something else. So the cells respond to the fenformin treatment by regulating another pathway. So they cannot regulate. So they increase glycolysis, and you can see this quite nicely on this slide. So both cell lines will increase glycolysis as you increase the dose of fenformin, and that's why the ATP levels stay high and the cells carry on growing at the normal rates when we do that. So just to finish off really, so bioenergetics of tumours is an area that's really developed over the last 10 years. The Warburg effect, which was in the 1920s, was first identified as aerobic glycolysis was the driver of cancer metabolism, but recently we've found out a lot more about cancer and tumours. And tumours can either show the Warburg effect shown on the left-hand side, there's a lot of in vivo work showing this occurs, but they can also be more oxidative. So the data on the right of the slide suggests other substrates such as glutamate, fatty acids, amino acids can also go into the TCA cycle. Or you might get something in the middle, so you get a truncated Krebs cycle with various things going on. So the cells can adapt, and that's something we've shown quite nicely on the previous slide. And understanding these levels of adaptation is really important in developing therapies. You can't just target one pathway. You need to think about what are the cancer cells going to do if we hit this pathway. What second pathway might they use, and can we block them both simultaneously? A nice review by Aubrey and Rossignol is definitely worth a read. It's just come out that details some of this. And what's interesting is that more and more cancers are being looked at in situ. We can now do this. There are the tools to do this, the carbon-13 labeling, microarray, gene expression profiling. What we're finding is that many cancers now, brain, lymphoma, liver, lung, have different types of metabolisms going on in those cancer cells. So some are showing aerobic glycolysis and some are showing OXPHOS. And the recent work by Rossignol, it's not quite published, has managed to find markers that can actually show which cells are doing which. So you can actually then profile a patient and find out what combination of the cells are doing glycolysis and what are doing OXPHOS. And maybe that's going to be really useful in going forwards in terms of therapies. So just to finish off really, just to give you a few thoughts really, of how our work in vitro might relate to what goes on in vivo. So you take a tumour, it's got a good vasculature, you're going to get a gradient of glucose and oxygen across that tumour. And the idea at the moment is that the tumour is predominantly glycolytic, but what I'm saying is that maybe that's not quite true, and you could have regions of high OXPHOS or high glycolysis. So if you've got a higher glucose on the outside, maybe the outside of the tumour near the vasculature is very glycolytic, and as you move towards the core, you get more OXPHOS going on. We've shown quite nicely that as you drop the glucose, you can drive OXPHOS. We also have shown in other studies that even 2% oxygen is enough to drive respiration, so cells can respire at very low concentrations of oxygen. And because the top layer is glycolytic, the oxygen is going to diffuse through that quite effectively. So you will have a blue layer shown here doing OXPHOS, and towards the middle, when the tumour becomes more necrotic, there's not a lot of substrates, and the tumour will die. So if you add a compound such as Avastin or something that can prune away the vasculature, then you're going to change this environment quite significantly. So you might reduce the glucose, you'll reduce the oxygen, so the periphery of the tumour is no longer exposed to high levels of glucose, and as we've shown in our in vitro work, that here you're going to push the cells to maybe use their mitochondria, so you're going to generate more blue areas, so the OXPHOS is going to be the driver, some glycolysis on the periphery, but the necrotic area is going to increase, so there's going to be less nutrients getting to the middle, so you're going to shrink down, well not shrink down, but you're going to kill the centre of the tumour. If you come in then with a drug such as dichloroacetate, then what you might expect to see there is you're going to really push those cells to do OXPHOS, so the glycolytic cells on the outside will be driven to use OXPHOS. You're going to push the cells that are doing OXPHOS in the middle to use even more oxygen, and that will reduce the oxygen even more within the tumour, and you'll get an even bigger necrotic core. A nice study by Adrian Harris's group, shown quite nicely at this last meeting, and they just published this, is that if you combine Avastin and DCA, you get a really nice synergistic effect in some xenograft models. Individually, the drugs don't do very much, but combined, they work really nicely. So this just shows that combining compounds is probably the way to go, and we need to work out what's going on in these systems to really target which pathways to hit simultaneously. And then from what I've been saying today, you might imagine at this point you don't have any glycolytic cells at all, and at this point the cancer cell is going to be really prone or really toxic, not toxic, sorry what the word is, mitochondrial inhibitors are going to be very efficient. So you can actually target the cancer cells with something like fenformin, metformin at very low doses. You don't need to get deep into the tumour. The living cells are on the outside, you've already killed off the periphery, and we think this is an interesting strategy to go with. So what we intend to do in the next couple of years is to try and develop in vitro models to look into this. To work under low glucose and low oxygen concentrations is quite tricky. You can do static cultures, but really they're not ideal. So what we're doing at the moment in collaboration with a Korean collaborator is to develop perfused models, so plate-based systems where we can grow the cells under controlled concentrations of glucose and oxygen and drugs, and monitor them over 3 or 4 days or a couple of weeks, and that's where we're developing. Good histochemical markers are essential if you're going to look at this. So as I said, the Rossignol group have found 40 markers in lung tumours could predict whether the patient tumours were OXPHOS or glycolytic, or at least isolate, identify those cells, and that's really important if we're going to try and take some of the work we're doing in the lab where we know what the phenotypes are to identify the correct drugs to use on the patients. The xenograft models is an interesting problem potentially with this. Mouse blood glucose is very high, between 6 and 10 millimolar. As I've shown you, these concentrations will drive the tumour cells to use glycolysis, but it's very different from what is happening in the human, where the blood glucose levels are much lower. So we need to be aware of that, I think, when we're using these models to look at metabolic compounds that target energy metabolism. Levels in the mouse can change with stress, If you're bleeding a mouse to do various experiments, the glucose levels can fluctuate markedly. And other issues, obviously cancer genetics, RAS, MYC, P53, they're all oncogenes that can affect metabolism. We really need to know what the status is of the tumours before applying any treatments. Drug-resistant metastasis are all other areas to think about. So just to finish off, really, I'd like to acknowledge people involved. So Michelle, Georgina, and Yvette in the middle. Also my group, the various placement students we've had over the years that have developed these assays, which have been really useful. Luxel, who have been a great help in providing us with reagents, technical support, etc. Tim Clarity in chemistry, we just started our journey in NMR. It's a bit like learning another language. We're getting there, and we're getting a lot more data on the mass spec side coming through. And of course the Williams Fund, who funded this research, and without them we wouldn't have been able to do this work. So I'll stop there, and I'm happy to answer any questions.